A very good morning to everyone and welcome back to MEAB Science's channel of Dr. Deepankar Ghosh. Today we are going to discuss and share a scientific survey on COVID-19, which is a tasty bites of the short-term e-summer project done by BSc Microbiology and BSc Biotechnology students of JIS University, Kolkata. The entire combined project has been conceptualized and further designed by Dr. Deepankar Ghosh, who is the Assistant Professor in Microbial Engineering and Algal Biotechnology Laboratory, Department of Biotechnology, JIS University, Kolkata. So let's begin the journey of this project. So the first project is the COVID-19 Physiology and Mode of Transmission, which is being conducted by Onik, Mehfuz, Shumon, Saurabh and Shobhan. Those who are the BSc Biotechnology 4th semester students. What is COVID-19? COVID-19 is an acute respiratory disease caused by the novel coronavirus SARS-CoV-2, also known as 19nCoV. COVID-19 coronavirus SARS-CoV-2 belongs to the beta coronavirus genus originating from bats. Beta coronaviruses can infect the mammals, are zoonotic pathogen, and can cause severe respiratory disease in humans. Other viruses in this family are SARS coronavirus, MERS coronavirus. COVID-19 has approximately 79% sequence identity to the SARS-CoV and 50% with the MERS-CoV. In addition, the homology modeling shows the COVID-19 has a similar receptor binding domain structure as a SARS-CoV, which suggests the SARS-CoV-2 uses ACE2 receptor in human for infection. The structure of COVID-19 consists of following a spike protein, hemagglutinin esterase dimer, a membrane glycoprotein, envelope protein, and a nucleocapsid protein and RNA as seen in the figure below. The spike protein is heavily glycosylated, utilizes N-terminal signal sequence to gain the access to the ER and mediate attachment to host receptor. It is the largest structure and makes the distinct spikes on the surface of the viruses. For SARS-CoV-2, a S protein is cleaved by the cellular furin-like protease TMPRESS2 into two separate polypeptides S1 and S2. S also consists of receptor binding domain which binds to the viral receptor ACE2. Single-stranded positive-stranded RNA is the genome of the virus. The nucleocapsid protein binds to RNA in vitro and is heavily phosphorylated. The N protein binds the viral genome in a beads of the string-type conformation. The protein likely helps adhere the viral genome to replicase transcriptase complex RTC and subsequently package the encapsulated genome into viral particles. The envelope protein E is found in small quantities within the virus. It is most likely a transmembrane protein with ion channel activity. The protein facilitates the assembly and release of the virus and has other functions such as the ion channel activity. It is not only necessary for the viral replication, but it is also useful for the pathogenesis. The membrane protein M is also most abundant structural protein. It does not contain signal sequence, exists as a dimer in the virion. It may have two distinct conformations to enable it to promote the membrane curvature as well as bind to the nucleocapsid. Hemagglutinin esterase dimer protein HE is present in the subset of the beta coronaviruses. The protein binds sialic acid on the surface glycoprotein. The protein activities are thought to enhance the S protein mediated cellular entry and virus spread through the mucosa. There are three distinct modes of transmission which is being found in the SARS-CoV, that is the droplet transmission, contact transmission, aerosol-borne transmission. The droplet transmission occurs when the respiratory droplet are ingested or inhaled by the individual in the close proximity. Secondly, the contact transmission occurs when a subject touches a surface or object contaminated with the virus and subsequently touches their mouth, nose, or the eyes. Aerosol transmission is the final one, which occurs when the respiratory droplet mix into the air and forming aerosol and causes the infection while inhaling a high dose or aerosol into a lungs in a relatively closed environment. So this is a typical molecular mechanism of the transmission of the COVID-19. So it has been highly depicted by the Vilgeri et al. In Science of Total Environment, which has been published in 2020. They clearly depict that how COVID-19 start interacting with the ACE2 receptor and causes the respiratory distresses finally. Now, what are the targets for the COVID-19? Many organs in the body, including the lungs, kidneys, and the small intestines and the brains are rich in ACE2 receptor. So their widespread prevalence is one of the reasons why the scientists think the COVID-19 can wreak havoc throughout the body. So that is the end of the project 1. The next project deals with the genetic diversity of COVID-19. The project has been designed and organized by Amrita, Arpon, Debangshi, and Smriti. Genetic diversity of COVID-19. The assessment of genetic diversity among 86 complete or semi-complete genomes of the SARS-CoV-2 viruses revealed 3 deletions in the genome of isolate from the Japan, USA, Australia, in addition to many other substitution mutations. The deletion mutations were in the ORF1ab gene and at the end of the genome. Of the 93 substitution mutation and 42 changed in the amino acid sequence of structural and non-structural proteins. The 3 and the 24 nucleotide ORF1ab are expected to reduce the protein sequence by 1 and 8 amino acid residues respectively, without changing the reading frame, but the functional effects have yet to be investigated. The alignment of the SARS-CoV-2 reference S protein gene against all SARS-CoV-2 sequence genome from China, USA, Japan, Australia, and Taiwan revealed 99.97 to 100% identity with 100% query coverage, while the identity and coverage of SARS-CoV S protein gene were 74 to 5% and 91% respectively. Also, the S protein gene from the bat SARS and the SARS-like coronavirus isolates shared 76.5 to 83% identity with that of SARS-CoV-2. This agrees with the previous conclusions regarding the evolutionary analysis of SARS-CoV-2 in the phylogenetic analysis we performed. The SARS-CoV-2 virus were in the same cluster regardless of the geographical region. So these results strongly suggest the possibility of a recent common ancestor for all SARS-CoV-2 for transmission of the same virus strain across the countries. The next most interesting topic of this project is the literature survey on genetic comparison of COVID-19 with existing viruses and protozoal parasites and conspiracy theory on COVID-19. The project has been conducted by Sushovan and Ronojit, BSc Biotechnology 4th semester students. Literature survey on genetic comparison of COVID-19 with existing viruses and protozoal parasites. There are 2 subjects are there. One is what are the genetic comparison of HIV virus, malarial parasite, MEAB, corona, and novel COVID-19. Secondly, the conspiracy theory that COVID-19 is a man-made or artificial virus. These questions are the major concern of this entire project. Literature survey on genetic comparison between COVID-19 and HIV. Number 1, genetic material of both viruses are same. Both are RNA containing viruses. Both live spikes in their outer surface. By the help of the spikes, they attach with the receptors and enter to the body. Researchers provided evidences that the COVID-19 can infect T lymphocytes, the same cells targeted by HIV. COVID-19 may exhibit lymphopenia or an typically low number of lymphocytes in the blood like HIV causing immunosuppression. They both mutated very rapidly, constantly changing its appearance and remaining a step ahead of any response produced by the body. Both entering in a cell integrates into the host cell's genome, effectively making itself invisible to the host's immune responses. The spike glycoprotein of the COVID-19 are 100% identical with glycoprotein 120 of HIV-1. So another literature survey has been done on genetic comparison between the malarial parasite and COVID-19. So it revealed that there are no any such similarity between them. Secondly, malarial parasite is a protozoa, and in other hand, COVID-19 is a viral regime. The genera is completely different. Now the question is, if there are not any such similarity, then why we use hydroxychloroquine in the treatment of COVID-19? It's a very big and challenging question at present scenario. So literature survey says that while we are comparing the genetic comparison between the malarial parasite and COVID-19, the entire mechanism of action of these anti-malarial drugs would result in an increase in endosomal pH by preventing the fusion between the virus and the cells, or interfering in the glycosylation process of the cellular receptors of the coronavirus. In addition, it is hypothesized that these malarial compounds have an immunomodulating effects that contribute to increasing their efficacy in vitro against the COVID-19. Here, Dr. Ghosh has also included some new insights on the molecular mechanism and the target of the repurposed drugs. So this is a nice new insight on the antiviral effects of the chloroquine against the COVID-19, which clearly depicts here that the chloroquine may have some effects on the COVID-19 following few points. Like cell surface receptor binding, it may interfere with the endocytosis of the viral particle, and it can also interfere with the viral transport. It can inhibit the viral uncoating. It can interfere with the viral transcription, translation, including post-translational modification of the viral proteins, and followed by the assembly and budding of the complete virion particle of COVID-19. So this is a nice molecular mechanism of antiviral effect of chloroquine, how it actually works against the COVID-19. Cellular and molecular possible sites of action of chloroquine and hydroxychloroquine as an antiviral agent. In this pictorial presentation, X represents the site of inhibition by chloroquine or hydroxychloroquine. Chloroquine or hydroxychloroquine inhibit the viral binding to its cell surface receptor. This antiviral compound, antimalarial drug, inhibits the sialic acid biosynthesis through the suppressing quinone reductase 2 activity which affect AC2 receptor activity. Chloroquine or hydroxychloroquine inhibits the virus pH-dependent endocytosis through increasing the pH, and it interferes with the viral uncoating. Even it interferes with the assembly and budding, leading to the accumulation of the viral vesicle within the trans-Golgi network. Chloroquine interferes with the lysosomal protein degradation and lysosomal fusion with autophagosome formation. Hydroxychloroquine can interfere with lysosomal activity and prevents the major histocompatibility complex 2 expression level. Finally, the chloroquine interferes with the TNF release and binding from the macrophages and or to monocytes. Chloroquine inhibits the phosphorylation of the p38 MEAB and caspase in the T helper 1 cells, which in turn inhibits the pro-inflammatory cytokines production and virus replication. Hydrochloroquinone blocking of MSC expression prevents the T cell activation and expression of CD145 and cytokine release. Hydroxychloroquine impairs the TLR signaling through the increasing endosomal pH and interfering with TLR7 and TLR9 binding to their DNA or RNA ligand, thereby inhibiting transcription of the pro-inflammatory genes. And finally, hydroxychloroquine inhibits the binding of DNA to the cGAS and therefore reduce the cytokine transcription and production, where the ACE2 is angiotensin converting enzyme, MSC represents the major histocompatibility complex, TLR represents toll-like receptor, cGAS represents cyclic GMP-AMP synthase, and MEAB is mitogen activated protein kinase. So this is another tentative molecular mechanism of remdesivir for the treatment of COVID-19, which conventionally used for the HIV treatment. The probable molecular mechanism of remdesivir against the SARS-CoV-2, figure A, represents the diagram shows the SARS-CoV-2 viral entry and its RNA synthesis, which can be blocked by remdesivir. Detailed molecular mechanism of remdesivir to inhibit it also the synthesis of viral RNA molecule. Now, in summary, the race to find the COVID-19 treatment accelerates. It shows monoclonal antibodies and convalescent plasmid can also be used. Chemostan mesolites can also be used, which can interfere or impair the TMPRESS2 and AC2 interaction with the SARS-CoV-2. Secondly, lopinavir and ritonavir can also interfere with the polypeptide chain synthesis. They enhances the proteolysis of those polypeptide chains. On the similar way, remdesivir can also interfere with the RNA-dependent RNA polymerase and interfere finally with the replication and transcription complex formation in SARS-CoV-2. Chloroquine, hydroxychloroquine, as we discussed in the previous slide, that it interferes and inhibits the endocytosis process. So these are the possible treatment of different conventional drugs that can be also repurposed against the treatment of COVID-19. So these are the future tentative drug targets against the COVID-19. That means this diagrammatic presentation will give you some idea what should be the probable target of COVID-19, and it will accelerate the discovery of new drugs. The next concept is the conspiracy theory that COVID-19 is man-made or the artificial drug. A news report quotes some special speech by some country's highest authority that the corona is laboratory-made virus based on its own intelligence agencies. Earlier, that country's Nation Intelligence Director's Office said it was still investigating how the virus began to spread. But the office said it had determined COVID-19 was not man-made or genetically modified artificial virus. Accused country has rejected the lab theory and criticizes the investigating country's intelligence agency response to COVID-19. In 2008, Nobel laureate for the Physiology of Medicine from France, caught the media's attention when he recently endorsed a COVID-19 conspiracy theory. He said that the COVID-19 is man-made, and the scientist also argued during a TV interview with the French TV channel that the element of HIV-1 retrovirus, which he found in 1983, can be found in genome of the new novel coronavirus. He also said the element of malarial germ, the parasite Plasmodium falciparum, can also be seen in the viral genome of SARS-CoV-2. He also said, "We were not the first since a group of Indian researchers tried to publish a study which showed that the complete genome of this novel coronavirus has sequences of author's virus with HIV." In a separate podcast episode with a different outlet, the same Nobel laureate further said that the virus had escaped in an industrial accident from the accused country's city laboratory when accused scientists were attempting to develop a vaccine against the HIV. The Indian paper, titled "Uncanny Similarity of Unique Insert in 2009 nCoV Spike Protein to HIV-1, GP120 and GAG," which has been published by Pradhan in BioRxiv 2020, but it's not peer-reviewed, which claim to find similarities between the new coronavirus and HIV. Is completely based on the improperly selected data and statistical analysis method that violate the basic norms and incorrect argumentations. Similarly, another scientist, Young, a scientist at the Peking University School of Basic Medical Sciences, wrote in a blog post recently, and COVID-19 was previously known as 2019 nCoV. The world-eminent scientific communities and few scientists slammed the Indian study that fueled the coronavirus rumors. The finding, which led conspiracy theory that the new virus might be a bioweapon, have sparked a huge controversy. The multiple scientists, including the Ding, a science economist at Harvard University School of Public Health, and also Zhengli, a Chinese virologist at the Wuhan Institute of Virology and a member of the team that found the origin of SARS virus is bad, and have criticized the paper and as lacking of scientific merits and have called it misleading completely. And finally, the author withdrawn the paper from this specific reputed journal server. Innovation on corona detection kits is another important aspect during this COVID-19 outbreak situation. The project has been designed by Shwangi and Shan, those who are the BSc Microbiology 4th semester student at JIS University, Kolkata. These are the types of corona detection kit. Number 1, COVID-19 Rapid POC CIVD detection kit, a lateral flow immunoassay test to quantitatively assess the presence of an analyte from a patient sample or specimen. In this case, the analyte being detected are IgM, IgG antibody specific for SARS-CoV-2. The Feluda testing kit. The indigenous kit is CRISPR-based, works by combining CRISPR biology and paper strip chemistry. Cas9 protein is barcoded to interact specifically with CoV-2 sequence in a patient genetic material. The complex then applied to a paper strip, whereby using two lines, one represents control, second one represents test, to determine the presence of infection. The indigenous real-time COVID-19 detection kit is the third one, which uses quantitative real-time PCR master mix primer probe and RNA template, each of which is developed in the laboratory itself. The fourth one is being proposed by Dr. Ghosh while he participated a online proposal submission program, that is Samadhan, organized by MHRD Government of India, where he depicts that this particular kit can be used to detect the presence of infection depending on the protein biomarker, if present, visualized under UV radiation with the help of fluorescent protein tagged to them. So then what are the positive impacts of the corona detection kit on the community? Firstly, the availability of the kit. A widely available test kit would drastically change the strategy for dealing with the virus and change our daily life. Secondly, performance improvisation. The test kit are produced rapid and accurate results greatly aid the common mass. Increase in the rate of detection kit make the better the diagnosis, better the drastically change the strategy for dealing with the virus spread. Industrial activity can also be enhanced. The production and marketization of the kit would require active functioning of pharmaceutical, medical and distributional engineering as well as other industries. Better treatment can be achieved by improving the diagnosis with the results in better treatment effect of the mortality. The better treatment would help treat the patient's life. Boost in the moral. Improved healthcare condition provide people hope and adding to their moral. Finally, the economic balance is very important, which helps to accelerate the marketization of the cheaper and better kit would help the economic conditions of the country. Then what are the negative impacts of the corona detection kit on the community at this current outbreak scenario? Number one is the highest price, a major problem for all wouldn't be able to have themselves to get diagnosed. Less production, a scarcity of product might lead to unrest among the common mass. The counterfeit product, these would help and cause the major distress to common masses. Economic imbalance and distribution that import and import of testing kit would affect the economy itself. So next, a most important and interesting project that has been designed by Sampriti, Subodip, Shumon and Shaurabh, that is the bioinformatics based approach on the novel drug discovery. Drug discovery or the development is a complex, time-consuming and the resource-intensive process to find an effective and safe medication that would improve the length and quality of life and relieves the pain and suffering that requires the multidisciplinary expertise and innovative approaches. Now, this can be obtained through the progressive steps where bioinformatics plays a very impactful role. Number one, the genome sequencing and comparative analysis with viruses that led to past pandemic situations. Secondly, to find a new drug or the drug combinations from the registered drug can be database archives, which can adversely affect the similar viruses and found by sequencing and comparative analysis. So these are the major crucial steps which are involved on the novel drug discovery or development. These are the four important points: target identification, target validation, lead selection, and lead optimization. Target identification is the accurate identification of the drug target is a crucial part of any drug development process. Target validation is a crucial step in the drug discovery as it is the basis of completely new drug exploitation and exploration for the COVID-19, and there must be a strong association between a putative target and the disease of interest. The role of target in disease must be clearly defined. The failure probably in the clinical testing and approval phases can be moderated by the drug target validation. The identification of the lead compound, the chemical compound as a complement to the target that shows desired biological and pharmacological activities. The conventional methods, random screening of natural products and a chemical-based laboratory is carried out, which is much time-consuming and costly as well. So lead optimization is carried out to improve the potency, selectivity, and pharmacokinetic parameters. If a molecule has no toxicity, no mutagenicity, then it is considered to have potential for use as a lead molecule. Now, these are the very good impact of the bioinformatic tools, which are program-based, algorithm-based, plays a crucial steps involvement in the novel drug discovery compared to the conventional approaches, which is really very time-consuming. That means the bioinformatic tool can do these four important crucial steps for the drug discovery, but also it reduces the time and more efficient process. Now, number one, these are the different softwares which are involved for the target identification, like PDB, HMDB, KEGG, PubChem, T3DB, and CTD. These are involved for the target selection. Secondly, for the target validation, most important softwares or bioinformatic tools are useful. That is Proteome Analyst, ELM, and the ProtFund. For lead selection, ChemBank, PubChem, SciBile, Schrodinger, and the Dragon can be useful tool. Secondly and finally, the lead optimization can be carried out by using different bioinformatic tools like ADMET, Tripodins, and Acylrise, and the Krembi-Strops. This is the final segment of this project that deals with the impact of COVID-19 on agriculture, food safety, and pharma industry. The project has been designed and conceptualized by Bishwajit, Debojit, Debojyoti, Gaurav, Nazim, and Partho. They are the BSc Biotechnology 4th-semester students at JIS University, Kolkata. COVID-19 evidences have been increasing in a massive, faster rate, which is the growing major concern in countries like India and other countries as well. It is for sure it impacts harshly to the health sector. However, the complete shutdown of all economic activities except essential services will create an economic crisis and misery for the poor, with massive job losses and rising food insecurity in agriculture, food, industrial, and pharma sectors. The economic shock will likely be much more severe for India for two reasons. Firstly, the pre-COVID-19, the economy was already slowing down, compounding existing problems of unemployment, low incomes, rural distress, malnutrition, and widespread inequality was there. Secondly, India's large informal sector is particularly vulnerable. Out of the national total 465 million workers, around 91% were informal workers reported in 2017 and 18. Lacking regular salaries or income, these agriculture migrants and other informal workers would be hardest hit during the lockdown situation. Thus, it will make a big impact on agriculture, food safety, pharma industry due to the current COVID-19 scenario directly or indirectly. Agriculture and food supply chain. COVID-19 is disrupting some activities in agriculture, food supply chain. Preliminary reports have shown that the non-availability of migrant laborers is interrupting some harvesting activities, particularly in the northwest India where the wheat and the pulses are being harvested. There are disruptions in supply chain because of transportation problems and other issues related to it. Food prices have declined for wheat, vegetables and other crops and fruits as well, yet consumers are often paying more to buy it. Media reports have shown that the closure of the hotels, restaurants, sweet shops and the tea shops during the lockdown situation is already depressing the milk sales. Meanwhile, poultry farmers have also been badly hit due to the misinformation and particularly on social media that chicken are the carrier of COVID-19. In another way, the cost of drug and the raw material and supply chain in the pharma industries are highly and drastically affected. The COVID-19 outbreak is a very dramatic situation. We believe there would be indirect effects on the continuity of supply of medicine due to lockouts, manpower supply, raw material supply issues, and stoppage of transportations. There are few examples. Likely the cost of paracetamol in India has gone up to Indian rupees 400 to 450 rupees per kilograms from the rupees 250 to 300 per kilogram, while the price of the vitamins and the penicillin have also gone up by 40 to 50% in India after the COVID situation. If the current situation continues and goes on to prolong, the cost of essential drugs and amenities might increase as well in India and other countries all around the globe. The pharma supply chain is fragile as well, and impact created by the COVID-19 has brought into the limelight once again. Therefore, this is the time to let's join hands together and fight against the COVID-19 to attain a sustainable, greener and healthier India for our next generation. Thank you very much for watching.